All right, so this is going to be video on blood, and you see here the flat blood cell model that we have in the lab. And I'll point out that these small, represented by blue cells here along the model and here, those are going to be your thrombocytes or platelets. Obviously, these ones are your erythrocytes or red blood cells. And then we have the 5 different kinds of leukocytes. So type number 1, these here, which you can see have granules in the background, but they don't stain either red or black. These would be my neutrophils. So this is a neutrophil, these 2 are neutrophils, and these 2 are also neutrophils. It's one of the most abundant types of white blood cells. Next, we're going to look for red staining granules, and there are 3 cells on this model here, here, and here that represent the red staining. And if you remember from lecture, those are called eosinophils, and they stain that way based on acid stain. And then the most rare of the granulocytes would be the basophils. And so to represent that, there's only one basophil on this model, and it is right here. And they're the ones that the granules stain like a blue blackish color. Okay, so those are your granulocytes, the neutrophils, eosinophils, and basophils. Next, we have your agranulocytes, which are the leukocytes and the monocytes. Okay, so, excuse me, the lymphocytes. Lymphocytes and the monocytes. So monocytes are very large cells, like this one, and this one, and this one. The monocytes are the ones that start out in the blood, and then within 24 hours, they're going to migrate and crawl into the tissues. I'm not exactly sure why they chose to represent these as having a dark gray background. I don't really see that when I see them in the microscope, but that is how they're represented on this model. And then we are left with these 2 right here. Both of these represent lymphocytes. Now, lymphocyte is almost all nucleus with just a small halo of cytoplasm around it, whereas the monocytes are one of the biggest cells that we will see in the blood. This one is kind of small, just barely bigger than a red blood cell, unless it has been activated. And so here, this is supposed to be an activated lymphocyte. Down here, this is an inactive lymphocyte, and active lymphocyte. All right, so those are all the cell model versions of the blood. The other part of this is blood typing. And so when you're looking at these little cards, the thing to remember is you're looking for clumping as a positive indicator. So on these little things, A is here, B is here, and RH factor is here. So if you see the dots, that represents clumping or agglutination. That means it is positive for that antigen. So for example, this one is clumping in the A and in the B, but not in the RH. So this would be blood type A, B, negative. And not on the ones on the test, but the ones that we're practicing with in class, you can see written on the back, it says AB negative, okay? Here we have clumping, clumping, and clumping. So this would be AB positive. This one, the A is clumping, but the B and the RH are not, so this would be A negative. Here we have, whoops, this one's upside down, sorry. We have no clumping in A, we have clumping in B, and no clumping in RH, so this would be B negative. Over here, neither A nor B is clumping, but the RH factor is, so this would be O positive. This one would be O negative. This one's also flipped upside down. There we go, flip it around. This one would be B positive, and this last one would be B negative. So that's how we would read these plates. And there'll be some examples of on the test. You will not be able to pick them up and look at the back. They should not be labeled on the back for the test. So don't think you're going to get away with it that easy. But anyway, other than looking at the blood on the slides, that should be a pretty good covering of blood cells that would be represented on this next practical.